Hello everyone, myself Rajendra Shetty, Assistant Professor at Ganpat Parsekar College of Education, Harmal, Goa. Title of the unit, Alteration in Chromosome Number. Module name, Meiosis in Structural Heterozygote. Outline, Structural Heterozygotes, Types of Chromosomal Aberrations, Meiosis in different types of structural heterozygotes. Learning outcomes. Explains structural heterozygotes, understands types of chromosomal aberrations, describes meiosis in different types of structural heterozygotes. Structural heterozygote, diploid organism which is characterized by a homologous pair containing one normal chromosome and other chromosome with structural aberration is called structural heterozygote. This you can clearly see in figure 2. Structural aberrations. Every chromosome has a characteristic structure due to presence of specific types of genes, specific number of genes, specific sequence of genes. This you can refer to the given figure, in which segment of chromosome is shown with positioning of 3 different types of genes. When change occurs in any one of them, it is referred as structural aberration. Types of structural aberrations. Structural aberrations can be broadly classified into 2 types, that is, change in number of genes and change in gene arrangement. Change in number of genes can occur due to deletion and duplication, and change in gene arrangement can occur due to inversion and translocation. Aberration due to change in number of genes. It occurs by deletion or duplication. Deletion is loss of part of chromosome. It can be terminal deletion, as you can see clearly in figure 1, or it can be intercalary deletion, which you can clearly see in figure 2. In this, a middle segment of a chromosome is missing. Duplication is presence of part of chromosome in excess of the normal complement. It is clearly seen in figure 3, in which you see genes C and D are present as an excess complement in a chromosome. Aberration due to change in gene arrangement. It occurs in 2 ways, that is, inversion and translocation. Inversion, part of chromosome is detached and reattached in reverse order of genes. You can refer to figure 1, in which you see a normal chromosome with gene sequence A, B, C, D, and second chromosome with inversion, in which the sequence of BC is reversed and the sequence now in this chromosome is ACBD. Translocation, part of chromosome is transferred to non-homologous chromosome, or exchange of chromosome segments occur between 2 non-homologous chromosomes called as reciprocal translocation. This you can see in figure 3. Meiosis in structural heterozygote with terminal deletion. Homologous pair has one normal chromosome and other deficient homolog with terminal chromosome segment missing in this type of heterozygotes. You can see this in figure 1. During meiosis 1 at zygoting stage, the chromosome pair precisely gene by gene all along the homologous region, but in deficient region, the normal chromosome does not show any pairing. You can refer to figure 2. Meiosis in structural heterozygote with intercalary deletion. In this, homologous pair has one normal chromosome and other deficient homolog with in between chromosome segment missing. Refer to figure 1. During meiosis 1 at zygoting stage, at the point of intercalary deficiency, a buckling or roof develops in the normal homolog. You can see this in figure 2. It is therefore known as buckling effect. Such a configuration is called deficiency loop or compensation loop. Meiosis in structural heterozygote with duplication. Homologous pair has one normal chromosome and other homologue has additional chromosome part. You can see this in figure 1. During meiosis 1, at zygote stage, the chromosome-bearing duplicated segments forms a loop to maximize the juxtaposition of homologous region. This you can clearly see in figure 2. Meiosis in paracentric inversion heterozygote. Paracentric inversion heterozygote has one normal chromosome and its homologue has some part of chromosome with inverted gene sequence, but centromere is not included in inverted sequence. This you can see in figure 1. In prophase 1, during synapsis, inverted loop is formed in chromosome with inversion to maximize the juxtaposition of homologous region, which you can see in figure 2. Crossing over in inverted region produces a dicentric chromosome. In anaphase 1, it forms a bridge from one pole to other pole, which you can see in the shown figure. This bridge breaks up randomly along its length, resulting in fragments with duplication and or deletion. An acentric fragment is also formed, which usually fails to move to either pole. At the end of meiosis, half of the gametes formed are non-functional, 1/4 functional with normal chromosome and 1/4 with inverted chromosome, as shown in the figure. Meiosis in heterozygote with pericentric inversion. Pericentric inversion heterozygote has one normal chromosome and its homologue has some part of chromosome with inverted gene sequence. Centromere is included in inverted sequence, as you can see in figure 1. In prophase 1, during synapsis, inverted loop is formed in chromosome with inversion to maximize the juxtaposition of homologous region, as you can see in figure 2. Crossing over in inverted region results in deletions and duplication, as you can see in the shown figure. It also produces acrocentric chromosomes. Anaphase 1 appears normal, but two chromatids of each chromosome usually have unequal arms depending upon where crossing over occurs. At the end of meiosis, half of the gametes formed are non-functional, 1/4 functional with normal chromosome and 1/4 with inverted chromosome, as shown in the figure. Meiosis in structural heterozygote with reciprocal translocation. In prophase 1, during synapsis, characteristics configuration of chromosome pairing is observed in order to affect pairing of all homologous chromosomes. Typically, a cross-shaped configuration is seen in prophase 1, as you can see this in figure 2. Later, this structure opens into either a ring or a figure 8 structure. Figure 8 configuration. In anaphase 1, both normal chromosomes moves to one pole and both translocation chromosomes moves to another pole. This is called alternate segregation or disjunction. This you clearly see in figure 1. Two kinds of gametes are formed, 50% normal and 50% with translocation, as shown in figure 2. Ring type configuration. In this, one normal and one translocated chromosome moves to same pole and their counterparts moves to opposite pole. This is called adjacent segregation. Adjacent segregation is of two types, that is, adjacent one, in which adjacent chromosome having non-homologous centromeres moves to same pole, as shown in figure 1. Adjacent to, in this, adjacent chromosome having homologous centromeres moves to same pole as shown in figure 2. Adjacent 1 and adjacent 2 will form gametes which would carry duplications or deficiencies and as a result would be non-functional or sterile as shown in the figure. Therefore, in a plant having a translocation in heterozygous condition, there will be considerable pollen sterility. Conclusion: structural heterozygotes show lots of irregularities during meiosis, which may sometimes result in formation of non-functional gametes. These are the references. Thank you.